So I interviewed Tom. Tom, I interviewed you in January, I think, this year. And when I talked to you, I mean, you did all these wonderful surveys, these embalmer surveys, and finding out how many of these strange white new fibrous clots they're finding in their bodies. So when I talked to you, you told me that you know this embalmer Richard Hirschman, who has been going public, and there are very few embalmers who have actually been doing that. He's been going public about what he's finding, and he's sharing pictures. And so through you, I got to know Richard Hirschman, and then I did an interview with him about a couple of, 3 or 4 weeks ago, and he told me about his findings. We talked for a long time about everything. And both you and Richard mentioned that you're working with a group of scientists that are actually looking into what these clots are, what do they consist of, and how do they form. And both you and Richard, you shared a little bit of what they're doing. It was all really intriguing, but you said, we can't really talk about it too much because, the findings are not final yet. We have to wait until they're done. And well, in the meantime, a couple of weeks later, the findings are there. And Tom told me that Greg Harrison, who is here with us, is the lead scientist of this team of scientists who are looking into the mystery of these white fibrous clots. So welcome, Greg.
Well, thank you, Cornelia. It's a pleasure to be here.
And Greg is joining us from Sydney in Australia.
That's correct.
And first of all, I would like to know, Tom, how did you get to know Greg?
Through Richard Hirschman. When I was doing the surveys, I was in contact with Richard very closely during that time because he was the very first embalmer that I really contacted and talked to in length about what was happening and what he was seeing in the embalming room. And then that led me to do these surveys, Cornelia. And this is what's causing the urgency of this whole situation, because as I've told you in my latest survey that was completed in December of 2024, through the year 2024, this third survey, the embalmers are still seeing these white fibrous clots. Like, here's a sample right here.
Yep.
Here's a bigger sample right here. I mean, these things are serious. This, huge, and they're seeing them, Cornelia, in a shocking 27% of their corpses in this most recent survey. And they've been seeing them in about between 20 to 30% of their corpses the entire last 3 or 4 years. So that makes it even more urgent to find out what is going on here, because it makes you wonder, well, what percentage of the living people are walking around right now with these things possibly forming inside them? Because we do know from contact with vascular surgeons and people that work in what's called cath labs that they're seeing these in the living as well. So we've got to figure out why they're forming, how they're forming, and what's causing them to form. And that's where Richard introduced me to Greg Harrison. And Greg has done a sensational job over the last year of doing multiple types of tests in order to figure out what the composition of the clots are, and as you say, how they're forming. So I'll let Greg take it away.
Right. Exactly. And when I talked to Richard, he told me that he pulls these white clots, he finds these white clots in about 50%, across as an average. Sometimes it's, maybe 39, and sometimes it's 70, but it's as an average, it's way more than that. It's 50%. Okay. So Greg, first of all, tell me a little bit about you. Who are you? And how did you get into all this? And then tell me about this team of scientists and we get into everything that you have done and the results.
Thanks, Cornelia. Look, I'll try and keep it relatively concise. I just wish to add one more thing before I start, if I may, and that is I've also been in contact with New Zealand embalmers and also John O'Looney in the UK. And so and also Laura Jeffries from Canada. So we've got 4 embalmers, 4 different countries, all confirming they're seeing these white clots. And around about 40 to 50% of all the bodies that they are embalming seem to have them in various concentration. In other words, some of them are literally riddled with them, others are finding occasional ones, and some are finding sandy blood and coffee grounds in the blood. Okay, so how this all started for me? First of all, I'm not a biologist and I'm not medically qualified in any way. I am simply an organic chemist. I am a polymer chemist by trade, which means I made additives for plastics. Plastics basically are the building blocks of, polymers are the building blocks of plastics. And I design products that actually enhance the properties of these polymers to withstand things like weathering, weather resistance, and for chemical resistance. So it's a pretty specialized form of chemistry that we did. And I say we did because I'm now retired, but I ran a manufacturing company in Australia for 20 years, and we manufactured these in a small plant. We had our own laboratory systems, and we became very familiar with the analysis techniques that have now been employed on the white clots. We had to use all of these techniques during the 20 years of my running the company to design the products that we made for polymers, okay, or for plastics. So I'm very familiar with techniques, and I'll run through them quickly. There's a thing, and I'm not going to give you all the names of them, but it's ICP-MS that finds elements, and we have HPLC that finds different proteins. We have LC chromatography that actually measures amino acids, and then we have histology testing, in other words, where we stain the actual clots and we expose them to UV, and if they glow in a certain way, it means there's amyloid or prionic presence there. And then we have other techniques that are far more sophisticated. So I'm across most of these techniques. I am lucky enough that I do some consulting work, and I can't describe I cannot divulge who I'm working with, but I have access to 2 universities. And 2 universities, these are in Europe, by the way. They're not in Australia, and they're not in the US. They're in Europe. And we have technicians in these universities who are studying there. They're studying protein chemistry, and they've got access to NMR and some more sophisticated techniques. I tell you all that as a groundwork because I initially came across this by watching an Epoch Times video years ago where Mike Adams was describing the analysis he had done for Richard Hirschman, and he, on the screen, he actually put up an ICP-MS elemental analysis. Now, because I was very familiar with ICP-MS, I freeze-framed the screen, and then I had a look at what the elements comprised of. And there was an abundance of phosphorus, sulfur, carbon, sodium, and tin. Now, tin piqued my interest because, believe it or not, but in polymers and plastics, tin compounds are used as catalysts to actually promote and drive the polymerization reaction. So I thought, what is tin doing in a white clot? Supposedly, and I was quite skeptical that these things existed at the time. That skepticism now is totally gone because we have received the samples ourselves. I've seen them. They're abundant. They are literally abundant now. There is no doubt this is a real phenomenon, and sadly, it's being maintained. So after watching the video, Mike Adams, I then contacted my colleagues in Europe.
Let me just correct one question. When did you see this video by Mike Adams?
2022. I think Mike ran this early 2020, February 2022, I think it was. And Mike will know the details, but I was skeptical of the results. So at the time, one of my proteges was actually working and running an ICP in Europe. He was actually working in a lithium battery company, and he was running ICP day in, day out. So I said to him, look, if I get you some samples of white clots, could you digest them in the same manner and just run the same tests? And they did. And they came back and they did the test a number of times. And they came back and said, we agree with Mike Adams. It's got high phosphorus, high sulfur, high carbon, high sodium, high tin, and a lack. The big issue was a lack of normal blood marker elements. In other words, there was very little iron, very little potassium, very little magnesium, very little zinc in these white clots. So these were not normal, as Richard describes them, grape jelly blood clots. These were rubbery, white, elementally rich in phosphorus and elements that are not normally found at these concentrations in a normal grape jelly blood clot.
Right. So in a normal blood clot, you would find a small amount of phosphorus. Is that correct?
That's correct.
Right, but in these white blood clots, you had like a huge amount, way more phosphorus than in the red blood clots.
The normal blood phosphorus level is somewhere between 100 parts per million up to 1,000 parts per million. That's the normal range. Sometimes you can get a spike up to 1,500, but we found 4,900 parts of phosphorus. Down the lowest amount of phosphorus we found in a number of samples, we analysed 4 samples, was 4,400 ppm.
And you found that across all the 4 samples, all 4 samples had these high amounts of phosphorus.
I've got the data. I can't show you the certificate of analysis because it's stamped by the university professor and personally signed.
Doesn't there just happen to be phosphates and sulfates in the COVID and Moderna vaccines?
No, we haven't done. Well, sorry, I don't like to go there, Tom, because we haven't analysed any of those vaccines. I know others that have, and they're finding perhaps some different results of what we're finding. But we're just staying with the science of what we know based on the hard analysis that we've done on Richard Hirschman's clots, okay, and also on some clots out of New Zealand through Brenton Faithful, okay. So we're matching up New Zealand clots, US clots, and I was hoping to get some samples from John O'Looney in the UK and also some samples out of Canada from Laura. They've yet to arrive, but we would like to do the same tests. We just want to get to ensure that every country has the same elemental composition.
Right. To your knowledge, are you the only? So this team, the small team of people, you and these scientists in Europe, they are all in Europe, right?
Yeah.
Is that to your knowledge? Is that the only team in the entire world that is looking at these clots and analyzing them and trying to find out what's going on there?
No, there are others doing it, but they're not looking at it through the lens or through the prism that we're looking at through it. In the sense that Mike Adams is the only other person I became aware of that's actually run the ICP analysis looking for elements. Now, the beauty of ICP, inductively coupled plasma spectrophotometry, if it finds elements in the concentration that it depicts, they are there, and you can do all kinds of other atomic absorption and a whole range of other techniques that are slightly less definitive than ICP, and they will all find them. So we know that his results were right. And in fact, we were able to replicate them using 4 samples, and we ran the test ourselves twice, and we got the same results. So it's definitive as far as we're concerned. There's no argument. To the point, I have an access to another laboratory by another colleague who is studying protein chemistry. He's actually doing a PhD in protein chemistry. And I asked him to run a thing called HPLC, high performance liquid chromatography, okay? And that tells you what the proteins are. Proteins are abundant in the human body, as you know, and in human blood, the most abundant protein is albumin. But the second most abundant protein in bloodstream is fibrinogen. And fibrinogen is part of the natural wound healing mechanism that the body employs to repair damage, particularly of the endothelial layers, with the lining of the blood vessels. They all have natural phospholipids in them. And when they're damaged and become inflamed, that's where the fibrinogen, soluble fibrinogen circulating in the blood, begins to lose solubility. And it begins to literally deposit and condense on the very damaged site. It's a natural mechanism that we have. However,
right. and we need it, otherwise we would bleed to death, right? If we cut ourselves. So you have the fibrinogen, and then when you cut yourself or you have a wound, then I think, is it thrombin, the clotting agent, that will help to form fibrin?
Plasmin actually stops the thrombin, and there's also thrombospondin that is there as well. All these things work together as blood clotting agents, and they work in unison with each other, but the plasmin then comes in to stop the clotting mechanism once the wound healing is complete. That's how it all works.
So it breaks up the clot. The plasmin will come in to break up the clot.
That's exactly correct. And it also truncates and stops the reaction itself. Okay, that's part of it. But by dissolving the clot, and the body can then naturally remove it, the process is called fibrinolysis. In other words, it breaks down the fibrin that has been formed by the fibrinogen. The fibrinogen is actually naturally polymerizing or condensing into fibrin, the fibers. Okay, that's a normal pathway. So the HPLC analysis we did, there was a gentleman by the name of Dr. Arnie Burkhardt who had also done the same work on Richard white clots, I think 2 years before us. And he used a technique called MALDI-TOF. And MALDI-TOF is an older technique, is not quite as definitive as the technique we use, which is HPLC. And Dr. Burkhardt got over 300 different proteins in those white clots. He also noted he did some other histology tests on it with Congo red, and he determined there was amyloid present in his samples. Now, unfortunately, Dr. Burkhardt met an untimely death. His work has been suppressed. We haven't been able to get a copy of his analysis because I'd like to compare it with the analysis we have. In our analysis, we found 541 proteins, and they were all human. And secondly, we then had a calculation done by extending the analysis to determine the concentration of all these proteins. And the most abundant protein we found in our white clot is fibrinogen beta chain, followed by fibrinogen gamma chain, followed by fibrinogen alpha chain. And the ratios are something like 36% beta chain, 16% gamma chain, and only 4% alpha chain. So the whole lot makes up 56% of the white clot composition. On top of that, we found actin. Actin comes from damaged muscle layer behind the endothelial layer of the vascular system, which means by the large amount of actin we found in the clot is telling us there's huge damage being incurred by the vascular system. There were a whole lot of there was myeloperoxidase and a whole lot of other proteins. Anyway, the ratio.
Greg, can I just quickly stop you? Because this is all really great information, but I want to go back to, because there's a lot of information. First of all, I want to go back to what you said about Arne Burkhardt. You said that you tried to get a copy of his analysis and you couldn't. How is that possible? I mean, was that a published paper, or why couldn't you get a copy of the paper?
Yeah. Arnie Burkhardt had the same problem we've got. The people who supposedly conducted his MALDI-TOF work did not want their identities released. And we have Dr. Ryan Cole, I think, has had some success trying to locate some of these people to confirm his results. But at the end of the day, the results were not exactly the same reason I've got, where the professors who have done the work using their MALDI-TOF equipment don't want their names out because they're frightened of the blowback that may occur to the universities. This work is highly controversial, okay? And
yeah, right. And yeah, go ahead. Sorry.
Unfortunately, but because in the early days, the mainstream media tagged it as being conspiratorial, it's been a very hard slog to try to confirm and prove, one, these clots are real, which Tom has certainly proven by his multiple surveys. Two, we know by the physical samples we have. And the latest set of analysis, and I have to be a little careful about this, we've just sent 14 of these samples to a laboratory. 14. So there's not a shortage of these samples, I can assure you. They exist. That's been our biggest problem.
Okay, so the other thing I wanted to ask you, it's about the hundreds of different proteins you're finding in these white blood clots. You mentioned 300 different proteins in the clots, and then you also said 541 that you found. 541 different proteins. So what's just to give me an idea, how many proteins do you find in normal, like in the red blood clot?
That's been a problem. We haven't been able to obtain a normal grape jelly blood clot to run a standard. And this is part of the problem we have. We would dearly love to get from an unvaccinated person a normal red blood clot, grape jelly, in other words, not actually dried and crispy, but in the same condition, preserved maybe in formalin. The problem you have is when you can preserve it in formalin, it destroys the hemoglobin. It leads to that bright red, beautiful shade to end up as a dark brown lump. and it changes the protein chemistry of that sample. If we preserved the normal red blood clot under argon and did the same analysis that we've just done, so it didn't contact formalin, we'd have a good chance of getting a standard HPLC of a normal red blood clot. Having said that, I found literature. There are papers published. There's been other analysis done, and they give us the levels and the ratio of fibrinogen. And the ratio in a normal red blood clot is 1 to 1 to 1. And the level in a normal red blood clot is about 20%, 25%. It's nowhere near the 56% that we found in the white clots, and it's certainly not dominated by fibrinogen beta chain. Okay, so we've had to use, and I will say we've used a bit of AI. We've actually got 3 AI engines working with each other. They do make mistakes. We do pick up errors. We don't trust what it tells us, but it's a good source, and particularly one in particular is very, very good at finding peer-reviewed academic papers. And I set a task sometimes to go and find me 10 papers, and it'll come back and say, I'm sorry, I can only find 8. But then it gives you the live links. So you go, some of them mainly paywall, many of them are available on the NIH or whatever. So we use this other engine as a validator to confirm that what AI has told us. But we don't rely too heavily. We rely more on the empirical analysis that we're doing because we know it so well, and it's hard data. You've got to be very careful when you use AI because it will mislead you. Okay? And that's why you've got to have secondary, and in our case, 3 engines that have access to the various data mining to validate what it's telling you. And on 4 occasions, I've proven it wrong. And on 4 occasions, when I presented it with the data and the paper and the dates and the peer review, it apologized.
Now, Greg, the HPLC analysis was very valuable to you, right? Because, like you said, the natural ratio of the fibrinogen alpha, beta, and gamma chains is a 1 to 1 to 1 ratio in normal blood in clotting, but you found it in a bizarre 9 to 4 to 1 ratio. That 36 to 16 to 4 ratio reduces to a 9 to 4 to 1 ratio. So you knew right there and then that there was something really wrong that was going on with the conversion of the liquid fibrinogen into the solid white fibrin. Something was interrupting or altering that process.
We did, Tom, because what was still fresh in my brain was the ICP analysis with the high level of phosphorus. So we did some further research and found of the fibrinogen monomer chains that exist, the beta chain, gamma chain, and alpha chain, the beta chain has the highest affinity to phosphorus, highest chemical affinity to phosphorus. So I set a computer, not AI, but a computer a task to sort out the concentrations of all the proteins that we found. And then I used AI to calculate the phosphorus affinity of each of the proteins. And the phosphorus affinity of each of the proteins for the top 36 proteins we identified were in the order of the highest to the lowest affinity to phosphorus. And at protein number 36, it loses its affinity to phosphorus, so other competing elements like sulfur and nitrogen, etc., begin to compete with it. It loses the affinity to phosphorus. So the top 36 proteins in our HPLC analysis, in the order of abundance, may I add, all have declining levels of affinity to phosphorus. That's not a coincidence.
So that validated your phosphorylation theory that phosphorus is...
That's correct. But then what had us puzzled was the amount of sulfur we found. We found 2,500 ppm of sulfur in these white clots. We didn't use ICP-MS because it doesn't pick up sulfur very readily. We use a thing called ICP-OES, which is a different type. And it found, and we did it twice, and we found 2,500 ppm of sulfur. Now,
and that's a lot of sulfur. I take it. That's a huge amount of sulfur.
There is another protein in the body called transglutaminase, which is a sulfur compound that is also freely available in the blood, and it helps cross-link the fibrinogen naturally in the wound healing mechanism to toughen the fibers. Okay, so when the fibrinogen is actually accumulating with the phosphorus site, it's cross-linking with transglutaminase. transglutaminase, one molecule of transglutaminase, 3 atoms of sulfur.
Now, there was a problem with sulfur in the past, right? And white clots. Can you tell Cornelia about that?
Thank you, Tom. Yes, that's precisely what I'm leading to, because in 1988 to 1992, there was a heparin white clot outbreak reported. And it's actually, there are papers in the NIH. I've downloaded them, peer-reviewed papers confirming white clot when heparin goes haywire. And they were using a highly reactive form of heparin, high molecular weight form, as an anti-clotting treatment for normal blood clots, thinking it was going to solve normal, and it did, but it generated white clots in 1988. So the papers all exist confirming this. I investigated the chemistry of heparin, and heparin is rich in sulfur. Sulfur and phosphorus sit side by side on the periodic table. They compete with each other. They have reaction sites called Lewis sites. Phosphorus has 6 of these, sulfur has 2. So phosphorus will outcompete sulfur in any kind of reaction. And what we're saying is if the heparin white clots that were found in 1988 had it been analysed using the techniques that we've employed, they would have found high levels of sulfur. And more importantly, they would have had a similar rubbery texture to the white clots we're seeing now. Now, that white clot incident in 1992 disappeared because they stopped using high molecular weight heparin. They changed the chemistry to a lower molecular weight, and I think they're using a heparin sulfate. And heparin sulfate means that the sulfur is more readily bound. So
right, and Greg, they made that change because patients developed these white blood clots, right? That was the reason that they withdrew the drug or changed the drug. And also, just to point out how important this finding is, or something that we already knew. I mean, this drug heparin that contained the sulfur. So sulfur has similar bonding properties to phosphorus, right? As you said, they sit right next to each other in the periodic table, so they have similar bonding properties. And the sulfur, we know from heparin, the sulfur binds with a fibrinogen. Is that correct?
Correct.
To form this weird cross-link thing that then ends up in these structures that are pretty much indestructible. These fibrous things that you cannot, that plasmid and nothing in your body can take apart again. Knowing this, and knowing that sulfur and phosphorus are so closely related, how is it even possible to put these phospholipid nanoparticles into a vaccine? Wouldn't you think, well, wait a minute, maybe we shouldn't use phosphor because we know it's close to sulfur, and we had this very strange occurrence with the white blood clots, and we had to withdraw the drug. Isn't that a reason to pause? And what do you think?
Okay, we think we've identified there are a number of things that provide phosphorus to this pathway. There's not just one. And unfortunately, the choice of the phospholipid nanoparticle construction, where you've got a phospholipid, a thing called DSPC phospholipid, it's part of the nanoparticle structure. And that phospholipid is also accompanied by some amine lipids or amino-type lipids as well, and some pegylated lipids. Okay, I'm sorry to have to use these terms, but it means it's actually a jumble or a mixture of lipids, but in that shell is a large amount of DSPC phospholipid. So it's carrying phosphorus. What we found, and there are a number of scientists who are well versed in this, who actually design the lipid nanoparticles, is that when the lipid nanoparticle breaks apart, remember the lipid nanoparticle is carrying the methylcitruridine core that is in the mRNA vaccinations. Okay.
So it's like a cocoon inside. It's like inside this slippery cocoon that allows it to go enter into the cells.
I like to think of it as a Trojan horse carrying the mRNA inside it.
Yeah, right. But a very slippery horse. I mean, a very smooth horse that is able to get into these cells because it's this lipid. It's this fatty lipid. Mm-hmm.
In one injection, remember, of the lipid nanoparticles, you have I think it's calculated with 1.5 billion or trillion of these particles. Anyway, I don't want to go into that. There's actually some disputes going on about how many particles there are, but there's billions versus trillions of them, okay? So my point is, when they go into the bloodstream, the lipid nanoparticles, when they release the methylcitruridine core in the bloodstream, they break apart. At that moment of breaking apart and releasing the methylcitruridine core, the phospholipid core becomes exposed. Exposed phosphorus heads in a fibrinogen-rich, soluble fibrinogen-rich plasma. It'll begin to bind with the available fibrinogen that weren't natural wound healing mechanism. Now, that's only initiation stage. Unfortunately, when the body then begins to generate the spike protein itself has some glycans coating the spike surface that have high affinity to phosphorus. And the spike protein itself has somewhere between 25 and 150 phosphorylation sites. Okay, so the spike itself will become phosphorylated in the bloodstream. So the more the body generates the methylcitruridate core activated spike generation, the more phosphorus will be gained in the bloodstream, courtesy of the spike protein.
So that's like some sort of toxic synergy going on here with these two guys
correct.
coming together.
Also, remember, I talked to you just then briefly about the glycans that are coating the spike protein. And this gets a little technical because there's natural spike and there's the spike that's generated by the mRNA vaccinations that are open conformation versus closed conformation. When you catch a natural case of COVID, the natural spike is in a closed conformation. It's like a bud, okay? Think of a rose plant. But the open conformation of the spike that's generated by the mRNA vaccines are open conformation. It's like a rose. So if you think of a rose bud as a natural infection versus a rose in your bloodstream, and with the opening of the configuration, you're exposing more of these glycan materials. Now, the spike is coded in a thing called GLCNAC, GLCNAC. Two types of GLCNAC. There's an O-GLCNAC and an N-GLCNAC, but they're scavengers. The O-GLCNAC in particular is a scavenger of phosphorus. And in the body, also in the bloodstream, is ATP, adenosine triphosphate, floating around in the blood, naturally phosphorylated with one available phosphorus for this material to hook up to. Okay, so the spike, because of the way it's designed. Let me just run through the glycans that are present, if I may. GLCNAC, get it right, galactose, mannose, fucose, and sialic acid. Now, the most abundant glycan on the surface of the natural spike is GLCNAC. GLCNAC just happens to be abundant in fungal species as a very toxic fungal species, a thing called Candida albicans, which we've studied. And just by coincidence, in 2012, in the Mojang mine in China, there were a number of miners who were shoveling out bat guano, became very, very ill, and some died. And so we did a little bit of research and thought, I wonder how much Candida albicans might be in bat guano. Quite a lot. And there's other fungi there that all have GLCNAC in abundance in them. That's a coincidence.
You think there might be a link between that possibly and what was going on at the Wuhan lab of virology?
It's all coincidence, isn't it? As Dr. Campbell would say. So anyway, we don't want to dwell on that. What we want to get to now is a mechanism by what's driving the white clots. So with this abundant generation of phosphorus components, you've still got a natural phosphorylation pathway happening. But that's driving the condensation of the fibrinogen, and then you get the generation of microclots. That's why we talk about sandy blood and we talk about coffee ground appearance of blood. And there's a well-renowned scientist in South Africa. Her name is Dr. Risia Pretorius, and she has a number of peer-reviewed papers accepted describing the conformation of amyloid microclots. Okay, these papers are readily available. I have copies. I've read them all. And she had confirmed by some proteomic analyses how to determine that these what microclots were in fact amyloid.
So before you, Greg, tell us quickly, what is an amyloid?
Amyloid is basically it's a misfolded protein that is very resistant to fibrinolysis, in other words, being broken down by the body. It's normally found in slow developing diseases like Alzheimer's, for example, and some of the prionic diseases, for example, Creutzfeldt-Jakob, for example, mad cow disease. So it's a very stable, naturally generated protein, but it's misfolded. When proteins misfold, they undergo a range of changes that prevent it from performing its original nature-designed purpose or process, okay? I'm getting a little bit tongue-tied here because of the complexity of what we're trying to describe. So it's the misfolding of the protein that is a problem.
Okay, so the amyloids are, if I understand that correctly, the amyloids are protein polymers that occur everywhere, right? Not just in that they occur naturally, not just in humans, but also in other living systems, like in.
It accumulates in tissue. Remember, amyloid is not normally seen in the blood. It's never been seen in the blood before. It's seen only in the brain at the accumulation of things like alpha-synuclein, tau tangles, a whole range of proteins that make up amyloid, but they all contain misfolded proteins. Okay.
Oh, so they are only in the brain, the amyloids.
Only in the brain. That's correct.
Okay. And the prions are a subcategory of some of the amyloids are prions. It's a subcategory of the amyloids?
All prions are amyloid, but not all amyloid are prions. That's exactly correct. Okay. So we did some analysis that has confirmed there is amyloid present in the embalmers' white clots. And again, the same phenomenon that Rita Pretorius has published in peer-reviewed. We use a similar pathway of analyses in the embalmer clots, and we've come up with exactly the same result that she came up with on the microclots. So the microclot formation and the macroclot result in the embalmers are one and the same pathological process.
Okay, so two questions here. So first of all, you said that the white clots contain amyloids, but they are not pulled out of the brain. They are pulled out of other body parts.
Well, yes, the white clots appear to contain a high level of an active amyloid. And with evidence is, first of all, with some histology staining where we stain the white clots with Congo red and we expose it under UV. Our initial test did not show fully formed beta sheet amyloid. We did not get what they call the bifurcation or the signal that we're looking for confirming fully formed amyloid, okay, which is fully formed beta sheets. What we got was a strange result, but it was still then the discoloration of bright pink that Dr. Barney Burkhardt used to describe. He said they'd found amyloid. We then applied thioflavin T, which is a slightly more definitive stain, and it has a higher affinity to amyloid precursors. In other words, the precursors have not fully formed the beta sheet structure of standard amyloid, okay? They are precursors. They eventually end up as the fully formed if the reaction is allowed to go to completion. Our analysis is telling us...
Does that mean they end up in your brain when they're fully formed?
Yeah.
So they will move into your brain?
The body generates amyloid naturally, and there are good amyloids too. The body needs amyloids, okay?
Right.
But it's the accumulation in endothelial level and narrow vascular passages that is a problem. And that's why it's a very slow, natural progression that does occur with people, sadly, who suffer Alzheimer's and various types of dementia, for example. And Lewy body accumulation is also related to amyloid. So Our point is...
and my second question, sorry to interrupt you, Greg, but my second question, Dr. Pretorius talked about the microclots and that she found amyloids in the microclots, right?
Yes.
So does that, do you think that these microclots eventually merge into macroclots, or are they something really different from, they always stay microclots?
They are the initiation point that continue to grow to the macroclots to the embalmers are seeing. They agglomerate, they self-agglomerate.
So it starts as a microclot, many microclots, and I've seen the pictures. It looks like, as you said, like dirty blood or like coffee grounds. They're very, very small, but lots of them. And so you say that's the way it starts, and then they kind of coalesce into these larger structures over time.
They grow by agglomeration. In other words, they find an inflamed region on the endothelial. Remember, these have been generated in the blood as well via the spike protein. And where the inflammation is occurring is where the the condensation pathway begins. That's where the microclots themselves, which are traveling in the bloodstream, and some of them do lodge in the brain, causing ischemic strokes, as you would be familiar with that term. So it's like a capillary blockage. They will eventually.
That's like a very small stroke, like a mini stroke. The
Yes, like a
ischemic
mini
stroke.
stroke. That's exactly correct. Mm-hmm. But they will continue circulating in the blood till they then start to accumulate on the inflamed surfaces because of their affinity for phosphorus. Okay. As they grow, they literally can either break off or dislodge, or we think even accumulate per se in the blood, because I know some of these white clots are being found in the arterial system. Remember also that the embalmers have never seen white clots, or even normal red blood clots, rarely, in the arterial system. They're normally found in the venous system. But we have thoracics, so we have surgeons, not thoracics, we have a number of surgeons who are literally pulling them out of heart operations. These have been videoed, and these are living people. These people have survived the operation with these clots removed. But
what you're describing, I was going to say, what you're describing, Greg, really is a new blood-borne disease, right? Because he said typically Alzheimer's patients, the amyloid material collected in the brain tissue, but now you're finding it this new phenomenon in the blood itself.
Yeah, we are tentatively naming it as a blood-borne systemic amyloidosis. Okay, and the reason we
so
have
amyloidosis meaning forming of amyloids.
That's correct. However, by the strict definition, there are 36 known amyloids, okay, that have been established, all classified, all named, and they all occur tissue-derived. Remember, this is being derived in the blood, not in the presence of tissue per se. And you can argue whether blood cells, in fact, are tissue, perhaps. But my point is, they're forming in a totally different manner and mechanism and ending up with amyloid properties, amyloid structures. There appears to be a bit of a truncation based on our results, where they're not fully forming the fully formed beta sheet that is known and named in the 36 known amyloids. We believe this is an amyloidosis occurring in the blood that's never been seen before. We think it's systemic because it's been carried around the body. But worse than that, we have proven conclusively with the other techniques I'll talk about now, the Raman spectroscopy and the RT-QuIC work we've done, it is definitely amyloid. Okay?
Or prion nature, right?
That's right. So,
just to put that into perspective, that's an absolutely shocking finding. Right?
It's a terrible finding. It has really alarmed us. It's alarming the scientists who are finding this and working on this, and we are being so careful to double check and recheck and make sure that our calculations are correct. We're running standards. We're doing all of this work again to ensure we are correct, because the ramifications of this are enormous.
I want to say at this point, it's a darn shame that the FDA, the CDC, university labs around the world haven't done this already. Greg and his team have paid out of their own pockets to do this research, and the whole world should have been working on this problem 3 or 4 years ago when these clots were first discovered. Yeah. And isn't it bizarre, Cornelia, that the FDA, CDC, and all these universities around the world don't want to touch this. They know the clots exist, but yet they don't want it. They've not asked Richard Hirschman for samples. They're not doing their own analysis, to our knowledge. So it's perplexing, isn't it? The lack of
It's just disappointing, Tom. We've had a bit of luck because I've had access to some of this equipment. Yes, I paid for some of the testing myself. They're not cheap, I can tell you. But I don't care. This is a bit of a hobby for me, okay, because I'm fully retired now. The important thing is to have someone in the CDC or the FDA do this work. This work should have been done years ago when the embalmers started to report these clots, when they could provide samples. And I know for a fact Richard Hirschman has submitted samples to the NIH and I think the FDA, and neither of them have responded.
Yeah, exactly. I mean, he found his first blood clot in like the summer of 2021, I think.
Yeah.
And then he soon after started sharing them. But back to what you said, Tom, I don't even think it's bizarre, because everybody knows, all the scientists, everybody, and then the guys at these federal agencies, of course, under the Biden administration and also under the Trump administration, because they were just like rogue agencies anyway. They weren't under his control. So they all knew that this was forbidden territory. You are not going to go near there. It's a career-ending area that you better not step into. So that's why it's so great that people like you, Greg, you're retired. They cannot destroy your career. So you have the freedom, you have the ability to go into this mine, this area that is littered with mines, and just do what you do, because they cannot blow you up.
Okay, can I just make a comment, please, Cornelia? It's not me alone. I've got some very close associates who have become friends over the years. I actually employed a couple of them in my previous business life, and they're the guys over in Europe now. But we've also made contact with some very high-level scientists who are at a much higher level than what we are, because I'm basically, and I'll use the term, I'm a bucket chemist, okay? I'm an industrial chemist using and making large volumes of these materials. We've made thousands of tons of these things over the years. So I'm a bucket chemist by trade. I'm nowhere near the Kevin McKernan, Rita Pretorius's caliber, okay? These people are way above us, and they've got access to far more sophisticated equipment than I could ever get to. Okay, for example, I'd like to move on now to the Raman spectroscopy work and the RT-QuIC work, real-time quaking-induced whatever, that has confirmed the presence of the amyloid, because these are as trustworthy and definitive as the ICP and the HPLC analysis I've just described. Okay? So when Raman technology finds amyloid, it is there. When you then add RT-QuIC, which analyzes the amyloid seeds, in other words, the microclots, and it finds high resolution of it, it is there. Now, where it's getting a little scary for the scientists doing this work, first of all, to run the RT-QuIC costs them $5,000 per run.
Per run. Wow.
Per run. That's correct. And we've just
how many runs do you have to do?
We've done 3. We had to get 3 data points. We just burned 15 grand.
Okay, and that's not even very, very many data points. You probably would have liked to have like 100 data points or 500, right?
Were those all 3 separate samples, Greg?
Sorry?
Were those all 3 separate samples of the white clot?
No, sorry, that's not correct, Tom. No. The reruns were done off the one sample to make sure that that one sample was correct. Okay, we've got to remember we've got 14 samples over there. We're working our way through this, but you can imagine that, first of all, the time. I just want to talk to you about the attenuation time now of RT-QuIC. When an amyloid, if you take a, say, an Alzheimer's amyloid that's been extracted from the brain, someone has sadly died of Alzheimer's, they've sectioned the brain, and you've actually been able to obtain a piece of the amyloid material that's clogging their particular vascular region, and you take that, you actually cryo grind it. You actually make it into a dispersion where you grind it to a very fine powder. You disperse that into a saline solution, and then you can inject that into a range of different types of equipment for analysis. And you can do animal testing on it as well. The point is, on an RT-QuIC, it normally would take that naturally formed amyloid between 15 to 24 hours to develop strong enough signal for the RT-QuIC unit to map it and detect it, or detect it and map it. The signal we're getting with the white clots prepared in the same manner with the same technique is only 7 hours. The technician who was, well, he's not a technician, he's a scientist, he's a neuroscientist, but the neuroscientist who is running this work is absolutely puzzled at the rapid onset that he is seeing. That's why he's done it a number of times. He can't believe he's getting a 7-hour elution time on RT-QuIC that would normally take 15 to 24 hours.
So does that mean that it's sorry? Does that mean that it's some sort of a super nasty kind of amyloid? I mean that
pretty much, yeah, pretty much saying it's a
super potent. Yeah,
it's happening at a faster rate that's never been seen before. There's some
so that
further
makes
work.
it highly infectious and able to replicate quickly. Is that the conclusion?
Correct. That's correct. But more importantly, we're sharing this work with some other scientists who are going down a similar pathway. We're not just relying on one group of results. And we will have more results to talk about probably another month or two's time. But one thing I can say, based on all the work we've done, we are 95% sure we have amyloid proven in the embalmer white clots, and there's a chance there may be some prionic activity in them as well. So,
just as a precaution, by the way, last week, Cornelia, through my surveys, I've been in touch with embalmer associations all over the world, and I've sent out an email to all of those embalmer associations asking them to warn their member embalmers to make sure that when they're handling the white fibrous clots now, that they do so cautiously, because we're not sure whether or not they may be infectious, prionic in nature. So just as a precaution, make sure they're wearing their gloves, make sure that they sterilize their instruments, clean any surfaces that the white fibrous clots touch, after they remove them from corpses. And I've also sent a similar email to the vascular surgeon societies in the United States, Canada, UK, and Australia, New Zealand, to warn them as well, to warn their members, vascular surgeons and cath lab workers around the world. Same thing. Be careful into these clots, and then also disposal of them as well, right? And another thing it brings into...
Yes. Tom, that's an interesting question. I mean, if you look, so first of all, I would like to know what they are highly infectious, you say, these clots that contain these prions, right?
They're indications, right, Greg?
They might contain prions. So they are highly... We don't, okay, you don't know yet, but there's a good chance that is the case. So if you... Right, but you have but let's say there's a chance that they do contain these prions, or so if you touched one of them, they're highly infectious, you said. So if you touch them with your bare hands, what are the chances of getting infected with one of these prion diseases?
We don't know. We don't know at this stage. It's too early to go down that pathway.
I do know one of the embalmers that responded to one of our surveys said that he had formed white clots himself in his arm. So I don't know if that was because he mishandled a white clot, or because maybe he had a bad case of COVID, or maybe he took the vaccine. That's speculation at this point in time.
Yeah, but the second point you mentioned, Tom, is how do you get rid of these things? I mean, how do you dispose of them?
They should be incinerated.
In a safe way.
One of the things that scares me is one of the few phone calls I have with my whistleblower, who's an endovascular specialist and cardiologist from Jacksonville, Florida. I won't name his name. But when I asked him, hey, what's happening to these clots? He says, well, we're throwing them away. I said, you're not sending them all to pathology? No, we're not sending them to pathology. We're just throwing them away. So I know there's medical waste, but I'm hoping that they would be incinerated. I would think would probably be the safest way to dispose of them would be to actually incinerate the clots.
Well, you have to ask them what happens when they're thrown away. I mean, where do they end up? In a landfill? Or
hopefully not in the water supply.
Yeah, hopefully not.
Another PhD study, I'm afraid.
We know a lot of the embalmers probably, a lot of things go down that drain, don't they? And when they're embalming, the embalming table is set up. So as they're draining the blood out of the corpse and putting the formaldehyde in, you see a lot of the blood coming off the corpse, including the ones with coffee grounds, draining down a drain.
Oh, that's actually, you're right. So does that just go to the sewage system, into the general sewage
I think
system,
so.
I guess, right?
Yes.
Yeah.
See,
There's
that's
another.
why this should have been investigated 3 or 4 years ago. That's why the CDC, the
Yeah.
FDA were really remiss. And thank God we have people like Greg and his team, and that are actually doing the hard work and trying to, lead the way in terms of
Right.
determining what's happening here. And
Y-
this is now, they've laid down this is the bookmark for the FDA, CDC, and the universities to get off their butts and do their own similar testing using the same kind of equipment, and they can
That's
either confirm or refute what Greg and his team
Right, right.
have come up with, right?
And Greg, you mentioned how expensive it is to do one of these runs. What was it? The RT-QuIC or
RT-QuIC, yes.
yeah,
Real
so
time
it costs
taking is-
$5,000 per run. So, and they are not funded. They're not in any way, they're doing these tests on their own, right? They're not in any way involving their universities or any kind of research institutes.
I mean,
Okay.
are there no
Okay. So
rocket
they have a
rockets?
funding for, they need money. Okay. So anybody
Yeah.
who watches this interview has some money and wants to put it to some good purpose, these guys need money, because it would be nice to have additional runs and
I'm very
tests.
uncomfortable to even suggest that. We don't ask for any kind of assistance
Yeah.
financially.
And I haven't
Because
either,
we-
because we don't want to be accused
Yeah,
of being
but we-
grifters and wanting money.
Okay.
So,
Okay. Yeah.
That's
Oh,
all.
okay. Forget about
Yeah.
it. But yeah, I understand that
Forget about
totally.
it.
But
Okay.
but I just want to point out that you're doing this on your own dime. You're doing this. You're financing this yourself, and there's
Yeah, it's a
nobody of the state funded universities and research institutes that's helping you in any way, shape, or form. Correct?
I think they've overlooked a few things. I think that they've forgotten their fundamental base chemistry. They haven't employed the techniques that are simple to give them pointers as to where they should go with it. As I said, the fact we've now got to far more sophisticated tests, and certainly the work that Dr. Pretorius has come using advanced proteomics, she's using to a very sophisticated kit in her techniques, and she's finding the same. Okay. The problem is the universities are scared to talk about it. They're worried about their funding. They're worried about the government reactions. There's a whole raft of concerns that these tertiary institutions have with getting behind this work. And this is a problem. And because it had such a bad start with people describing it conspiratorially as being, as fake, and nanobots and all this other stuff that's come up, it's been a huge distraction from a real problem that's been identified.
we
Now,
kind
we're
of
just
got off
saying-
on the wrong foot with the Died Suddenly film. There was controversy about the film itself, but unfortunately, and that put a little bit of a shadow on the reality of the existence of these clots.
That's correct.
Like I said, I bored through that by drilling in on what one of the embalmers, Mr. Wallace Hooker, said. He gave that briefing at the Ohio Embalmers Association conference in Columbus, Ohio, back in October 2022. And based upon that, I contacted the Ohio Embalmers Association, and then found out from their now president, Mr. Woody Wilson, that he's seeing these white fibrous clots in his funeral home up in Marysville, Ohio, about an hour north of where I live. So then I've since then found several more well-respected men in the industry. Kevin Swire, who's the president of the Manitoba, Canada Funeral Service Association, seeing the clots. Chris Calvey, the president of the Pennsylvania Funeral Directors Association, also has been seeing unusual clotting in his corpses. So we've got prominent people, and like I said, now this whistleblower from Jacksonville, Florida, who's a doctor who's been pulling them out of living people for the last 4 years, as well as another whistleblower that Dr. Phil McMillan found. So we've got people that are well respected in the medical and funeral industries who are confirming the existence of these clots. So that's not the issue now. The issue is getting the FDA and the CDC and the universities to pay attention to it.
The issue is, if we are right, and I pray we're wrong, but if we've identified a new blood-borne systemic amyloidosis, then the world needs to know about this real quick, okay? Because there has to be some hard research and hard questions asked now about, one, what is causing it? Two, how do we stop it? And three, how do we treat it, okay? This has got to be addressed as a high priority now. because what we are talking about is fact, backed by hard analytical science. Certainly soon will be backed by animal studies.
And backed by the surveys that say that we're seeing them in 27% of corpses. This is not a rare phenomenon. These things are prevalent.
It's correct, Tom.
Yeah.
It's not rare.
Right, absolutely. So, Greg, you said question is what is causing it, but we already talked about the spike protein in the COVID-19 virus. So when you get COVID, and the spike protein as it comes in the vaccine, and that there is a crucial difference. And I want to ask you about and that the spike protein, the way it's the mRNA and how it's wrapped into these phospholipid nanoparticles, et cetera, how it's way more devastating to the body apparently than the COVID virus that you get naturally. I want to ask you about this. So do you think it is possible that somebody who is unvaccinated but had COVID, as a natural infection and got over it, it probably wasn't a big deal because for most people it wasn't a big deal, got over it. Do you think there's any chance that the spike protein in that case would have cross-linked with fibrinogen and polymerized into these non-dissolvable fibrin structures and did terrible damage to your body, or is that not very likely or even impossible?
There's some scant evidence being gathered at the moment about what they call exomal infection, that theoretically cohabitating with someone who's heavily vaccinated could possibly pass on the spike protein via fluid interchanges with their partners. Okay, nothing's proven yet, but there are some worrying signs, and you may have heard me talk about the open reading frames that we've been researching.
Yes, I want to. Yeah, right. Okay. I want to ask you about that.
And by the way, we have some survey data that may point to that, because in the first survey I did back at the end of 2022, 44 of the 179 embalmers that responded to that survey said they did see the white fibrous clots in 2020, the year that we had COVID, but no vaccines yet. But then the number of embalmers seeing the clots exploded in 2021. After the rollout of the vaccines. So that would indicate that it is possible to get the white fibrous clots from just the virus itself. Also, in my people's blood clot survey that I ran late last year in 2024, there were, of the 1,400 responses, 80 of the people said that their blood clotting problem started in the year 2020, not, the year that we had COVID without the vaccines. Now, I didn't ask
and Tom, were those white clots?
No, I didn't ask them to delineate which type of clots. So those could have been grape jelly clots. But then again, the numbers exploded starting in 2021, going forward when over 500 saw them. So there is some statistical data from the surveys that points to the possibility of the white clots forming because of the virus. And Dr. Peter McCullough, though, has said that he believes that you're getting a lot less of the spike protein from the virus, whereas when you take the vaccine, it's producing the spike protein in much higher quantities for a much longer period of time.
Well, yeah, because the mRNA instructs the body to produce the spike protein itself. I mean, if you get the virus, if you get the natural infection, then you get the spike protein and your body fights it, and then it's destroyed and it's gone. Your body isn't taught to perpetually produce the spike protein.
But I want to go back to what Greg, continue to what you were saying about the ORFs.
Yeah, I just kind of say, first of all, the indications are that the mRNA generated spike is more pathogenic than the natural spike. Okay. And in that, we've done some proteomic analysis looking at the various open reading frames that are evident. And this is highly technical stuff and outside of my pay grade, quite frankly. But we had to rely a bit on AI, but we're able to find papers confirming this. First of all, let's talk about open reading frames. What are they? They're groups of amino acids that have various functions, and they're classified in a way that they're quite long. Some of them can be literally hundreds, if not thousands of annotations. And each of these annotations is a letter, and it's a group of amino acids, okay, in a certain pattern. What we're talking about are open reading frames, ORF 1 to 10. They've all been published, theoretically, in the NIH. So all of the first ORFs are known. However, there are papers starting to come out about unpublished ORFs and calculated ORFs, okay? So we started doing some research on this site. And sure enough, there are a number of ORFs, ORF 11, which is past 10. 10, by the way, seems to be reasonably pathogenic via a ubiquitin pathway, which is a technical interruption, if you like, to a process essential in the body. So ORF 10 is known. But ORF 11, which comes from a zoster virus, ORF 19, coming from Candida albicans, and ORF 29, supposedly synthetic, have been identified in other non-viruses, in other words, in fungi. And as I said, ORF 11 has been found in zoster virus. Now, doing some
wait, Greg. What is the Phosphorus? What is that? You just mentioned. The term that you just mentioned. What is that? You said that they are found in something like zoster.
Zoster, ZOSTER virus.
Okay.
Herpes.
What is that? Herpes. Okay.
Herpes. Okay.
So what we're talking about here are these open reading frames. We're talking about basically, like they said, this is the layout of the genetic code of the virus, right? The SARS-CoV-2 virus. Right. And there's certain, each ORF, open reading frame, codes for either like a structural protein, like, the membrane of the virus, or the nucleocapsid, or it can also code for certain functions, right, to make the virus replicate faster, evade the immune system better. So these are basically the building blocks for that SARS-CoV-2 virus that Greg's describing, and it also is going to be linked into the vaccines as well. So continue, Greg.
Okay, and when you say, Greg, when you say they are unpublished open reading frames, what does that mean, unpublished?
Unpublished in peer-reviewed journals, but
so nobody knew that they existed.
Well, this is a problem. So there are certain things that do exist that have been discovered accidentally, and certain things leaked. And the things that have been leaked stack up, but we are starting to get now into a very murky area. I really don't want to go there because I'm reticent to cloud what we've just described with all the hard science by going down the pathway of discussing bioweapons research. Okay, I don't want to go there because I made myself a promise years ago I would not do this research. But unfortunately, the analysis findings are pointing to these major ORFs, the ORF 11, 10, 11, 19, and 29 being highly pathogenic. And unfortunately, we found the papers, peer-reviewed, describing the origins of where these ORFs come from. Now, it's just coincidence that there's none of these ORFs found in a SARS-CoV-2 sub-eco coronavirus. Okay, there's no ORFs of the description ever published in SARS-CoV-2.
That doesn't mean they're not there, though. See, I'll even though Greg's not going to speculate, I'll speculate that one could speculate that both the virus and possibly the vaccines were engineered, and certain things were added into the virus to make it replicate faster, to make it evade the immune system better, to make it actually intentionally cause blood clotting. There's been bioweapons research in the past done by the Russians, done by the United States, done by the Chinese, that have have been addressed in those particular areas. So it wouldn't be a far stretch to believe that SARS-CoV-2 was a man-made engineered virus in the Wuhan lab, combining all the greatest techniques that the Russians, Chinese, and Americans have developed over decades. So
yeah, I think that's pretty much clear that that's the case now. Just with.
But then in the open literature, you'll only see papers that like identify the SARS-CoV-2 virus from ORFs 1 through 10, and then it stops there as if that's the entire length of the code. And that's all. What Greg is saying is that there seems to be evidence that there's more ORFs in more open reading frames than what has been published in these published papers.
I just want to stop you there for a moment, Tom, because the comment you made needs to be looked at from another prism, and that is the ORF stop at ORF10. When Shi Zhangli, the WIV bat lady scientist, was describing the release, the very first presentation she gave when she released the SARS-CoV-2 genome, she stopped short of the furin insert on the genome. Now remember, if a furin insert had been found for the very first time in a Sarbeco coronavirus, it would have been world-shattering news in the world of virology. But she stopped short. Now, we're not suggesting there's a link here, but it's quite possible the research on the ORFs has stopped short.
Well,
So she knew that furin cleavage site existed, but she did not talk about it when she. So, and that's been proven to exist since then. So if it happened in that particular case,
it was US scientists who found the existence of the furin cleavage site, and then they also found the existence of the PCSK9 site, okay, which is a human high affinity site to LDL cholesterol. There's a whole lot of things that were found that could have been divulged at the time that weren't. For example, the Pradhan et al. Paper that found the HIV mimics or segments that had an uncanny similarity to HIV. Then you had.
what you're saying, Greg, is a lot of the papers that may have been produced, especially those produced by the Chinese, might be what's called limited hangouts. They might actually describe some of the problem, but they aren't describing the major part of the problem.
That's correct. So I just want to finish off while we're talking down this rabbit hole, that the ORFs, we've been able to group between ORF 19, go all the way up to ORF 100. And we've got them between ORF 20, ORF 40, ORF 60, ORF 80, all the way up. And I've been spending data mining time finding the origin of these ORFs. Many of them, as I said, have been calculated. Many have been theorized, but many also exist in other existing things besides Sarbeco coronaviruses. Okay, so the ORFs are not a figment of someone's imagination. And the last point I'd like to raise, there's been some interesting leaked ORF diagrams uncovered. And those ORF diagrams are quite specific on the function of ORF 11, ORF 29, and ORF 19. As the last final word on this, I don't want to go any further, is ORF 19 looked to be the most pathogenic ORF of all of them and found in abundance in the fungal species.
So you're finding these open reading, these strange open reading frames that are found in, some of them are found in fungus. You find them in the virus, in the corona SARS-CoV-2 virus. So that sounds to me, that sounds to me like these ORFs are man-made.
Now
how does it get in there? How would it get in there, this fungus stuff?
And the next question, Greg, is if they're in the virus, what are the chances they may be in the COVID mRNA vaccines?
They will be. We've actually done some and we know that ORF 19 and ORF 11 are highly transcribed into the mRNA vaccination spike.
You see the problem we have here, Cornelia? We've got these mRNA vaccines. They're still on the market here in the United States.
Yep,
for any
I certainly do.
Over 6 months of age.
Not all the ORFs, ORF 10, for example, doesn't go across, okay? We've been able to work out which ORFs go across with the transfection, and I can tell you now, the highest incident is ORF 19 and ORF 11, followed by ORF 29.
I watched the new series that is in the process of being released, Birth of a Plague, and I think you are involved in the making of this. I think both of you, right? Tom and Greg, you are involved.
Greg and Wayne Crouch from Pure Media.
It's very well done. And I think in episode 5, you talk about the open reading. It's talked about the open reading frames. And you talk about, I think, 11, 9, 19, 29, 3A, RFS 6, and all of them are terrible. They're like demons. They are there. Their function is to damage the body, to suppress normal healing functions or normal functionality of the body. Correct?
They seem to work in concert. Some of them are very benign. but certainly ORF8, ORF2A and 2B, and even ORF1 are not benign per se, but in combination with the other ORFs. Okay, there's a lot of information out there that has not been published, and we can't verify a lot of this, but there's a lot of support data in other papers coming from other
Greg,
organisms.
is this something that if universities around the world would actually start taking a very close look at this, could they deduce this, or is this even beyond their pay grade? Is this something that you'd have to go to bioweapons research labs, DARPA, that kind of thing?
Yeah, you'd have to. Like people like Dr. Stephen QuIC know this inside out. Okay, Stephen QuIC has just done an excellent paper confirming that Pradhan et al. HIV segments exist, and he's basically he proved that they were right. But it comes from the NSF side of the virus. Okay, it's the NSP side of the virus. But my point is, there are people out there like Dr. Stephen QuIC who have the ability, as does Dr. Buckles and Dr. McKernan, to drill down into this and just, as I said, hopefully prove or disprove what we're finding. I hope they disprove it.
So that means, when people get these booster shots, so let's say like 6 billion people around the world got at least one of these mRNA COVID shots.
5.5 billion, yeah.
5.5 billion. Okay, huge
billion.
number.
Two-thirds of the planet.
And a huge number must have gotten the 2 basic shots. And then I think it just tapered off. People don't take the shots anymore. I think quite a few people still took the first booster shot. And which one are we now on, the 9th or the 8th? I don't know. And I think the uptake is low, like 17% or so in the US, which is still horrifically high. But every new dose, every time you get another one of these mRNA COVID shots into your bloodstream, you just amplify the damage. I would think you would damage the ORFs, everything. You get the spike. Everything is kept. I mean, we don't even know how long the spike protein survives, even if you just got the 2 basic shots. We don't know that. But I would think that with every additional shot you're getting, you're making things worse and worse and worse.
Yes, we think there's a cumulative effect. Some of the calculations we're doing, you've got to remember that the masses that have been found in the various bodies, when the embalmer finds these clots, the clot hadn't just occurred in that one spot. Okay, it's either traveled there, it's accumulated there, but chances are if you did a proper autopsy on the body and opened up the vascular system, you would find more of it. Okay, so the mass that is there is grams. We're talking grams and grams of this material, not micrograms, not tiny amounts. There's large masses of it. Now, in order to generate that physical mass of material, the more, if we're right, the more booster shots you have, the more you feed this reaction and drive this reaction, because you are providing the monomers and the presence of the phosphorylation. You keep replenishing it. There is some indication that maybe, dare I say, that 5 of these things will pretty much guarantee a white clot. But that's it's conjecture. I can't.
And that was confirmed by Dr. Phil McMillan's whistleblower who works in a cath lab, same cath lab for 20 years. Says, and he does have access to all the COVID vaccination records of all his patients. Right. Says he's extracting anywhere between 3 to 10 of these white fibrous clots out of patients per week in his one cath lab alone. And he says 99% of the time when he finds the white fibrous clots, that person's had anywhere between 1 to 8 jabs, and the more jabs they've had, the worse the clotting seems to be. Yeah, I know that's anecdotal, but it jives with what Greg just said.
Yeah, we are living under this terrible omerta, right? This silence where people like these vascular surgeons, I didn't even dare to, or this vascular surgeon association, they didn't even dare to forward your very simple and quick survey to their members, and we don't have the data. We don't really know how prevalent this is, how many people find this. Because people don't dare to go there.
Yeah, we've got a good idea in the dead, but we don't know the percentage in the living. So.
Well, also there's a personal factor too. My family is all vaccinated. My daughter's vaccinated. And they had terrible problems with the Pfizer vaccine. My eldest daughter had myocarditis, and my second eldest daughter had pericarditis, both diagnosed by doctors, nearly hospitalised. They had terrible reactions to it. And this weighs heavily on me personally to have found what we've found, knowing that they've had a number of these vaccinations. We've also had infections here too. I've had COVID, dare I say it, and my children have had a number of times because they've got young children who bring it home. We don't know what the multiplier effect of this stuff is. So
you could be an unvaccinated person and had COVID multiple times, and that's what you're saying, Grant. You could be an unvaccinated person, had COVID multiple times, and maybe in a certain fifth or sixth battle with COVID, maybe then you'll start developing a white fibrous clot too.
Well, my wife's been vaccinated too. I mean, this is the nightmare if you start going down this rabbit hole, as I keep saying, that you get into. You don't want to go there. You've got to stay with the science. You've got to stay with what the evidence is telling us, and keep all this other noise out there. That includes nanobots and all the other stuff, which, despite the enormous amount of microscopy work we've done, we've never seen a nanobot. I just like to throw in and back Dr. McCann on that. That's in the white clot. I'm not saying they're not in the vaccine. I'm not saying they're not in the bloodstream somewhere, but they certainly don't appear to be in our white clots. But
the results you've gotten from your ICP-MS and your HPLC and your thioflavin T and your RT-QuIC testing, all of it,
spectroscopy, and now next the animal feeding. We just have to stay focused on what the science is telling us.
Right.
And all this other stuff may come to pass or may not. But one thing's for sure, Greg, we want to know what's causing this. Okay.
Right. So, Greg, I have a question for you that I'm sure is of great concern to many people. Do you think that these white fibrous structures will eventually form in all people who got these mRNA COVID shots?
We don't know. There's a short answer. We don't know. All we know is there was obviously a correlation starting to occur between highly vaccinated and the development of the white clots. Whether there's a longer-term problem, which may exist from exomal infection, we simply don't know. Okay, and I don't like to speak because we've got no data.
I mean, there's so many things. There's batch variation. We know from the Denmark study there was, like 5% of the batches seemed to be responsible for 70% or so of the severe adverse events. Right. So we know there was poor manufacturing processes going on there.
Yeah. Placebos are out there. We know that there was a whole bunch of placebos. Stephen Kirsch did some analysis, and I think he found 30 odd of his analysis didn't contain mRNA of any description. They were definitely placebos. So we know Pfizer, and I don't think Moderna or any of them have published how many placebos and where they distributed the placebos to.
Right. Also, a lot of the shots might have been duds. They might have had active ingredient at some point, but then, they kind of dropped the cold storage requirements in a lot of places. So, you might have had an active ingredient in a shot that then, warmed up, became a dud. And in this case, you actually want to be injected with a dud. You don't want the full strength stuff. The number of shots matters, was like we were talking about earlier. In the case of fibrinogen, people have different starting levels of fibrinogen in their blood. Normal adults have anywhere between 2 to 4 grams per liter of fibrinogen in their blood normally. So if you have a lower amount of fibrinogen, it might take more of the spike protein and this phosphorylation effect to actually, induce a white clot in your blood system. Blood type could be an issue here as well. We've taken a look at like type O blood versus type, AB. Type O seems to be the least susceptible by some to getting the clots. People have different overall strength in their immune systems, so that matters too. Level of exertion matters. The soccer players and stuff that were functioning on a lot of adrenaline, and there's a lot of blood pumping into the system quickly. They seem to develop side effects a lot more quickly from the vaccines because it's going through their system faster and faster. So there's all kinds of things here that could be. And Richard Hirschman likes to point out, well, in nature, when you get a bee sting, some people react differently, right? Some people, no problem at all. Other people go into a shock, from a bee sting. So there might be something, on an individual basis that might have a propensity to form the clots more so in somebody than somebody else. So,
Okay.
what do you think
Well,
about
I
that,
mean, what
Greg? With all those explanations, I think they're all factors.
Yeah, yeah. But all I just want to, I have to go shortly, so I'd like to maybe finish up if I may. And Cornelia, thank you so much for the opportunity to air all this. It's important we stay focused on our science and we don't let the darker side take over. And we certainly don't become or lose objectivity on what we're doing. But if we can encourage the various authorities to take a good hard look, to do the fundamental analysis that should have been done years ago, to actually replicate some of the work that we've done and do it correctly and objectively, we just want them to understand what it is we've found, because there is no doubt, and I'll use it, that tired old term, Houston, we have a problem.
Yeah.
Yes. And yes, and I want to end on a note of hope, because there are a lot, if this omertà if people would acknowledge the problem, there is so much that could be done. And I found a lot of things that should be done and should have been done years ago, but forget about that. That should be done now, and that can be done now. That would be extremely helpful. And so, for example, I just mentioned a few, and then maybe Greg, you can talk more about that, because just to tell people there are things that there are things that can be done. So, for example, first of all, there has to be, and I'm taking that from the paper you sent me, an immediate and thorough scientific recognition that this problem exists. There should be in vivo modeling in animal systems to understand better what happens, how dangerous it is. There should be expanded clinical surveillance for neurocognitive sequelae, so consequences that affected populations. And there should be an urgent public health inquiry into vaccine-derived spike protein persistence, so that we understand better how long does the spike protein persist, what factors influence the persistence, and the prion risks. These are all practical things that should be done, instead of people are just, oh my God, am I going to die of this? Instead, we would say, okay, just accept it, and now let's do something about it. Instead of closing our eyes and not wanting to see, not wanting to hear, but tackle the problem, because we have a huge problem. So what other things are there that should be done, Greg?
At the moment, there are people working on combinations that may stop the effect of the spike, okay? In other words, reducing inflammation. From where I sit, and not being medically qualified in any way, I personally take some combinations of quercetin, vitamin C, for example, NAC, curcumin. My daughters and associates are also taking it because they found it helped them enormously to recover from the effect of the vaccines. One anecdotal piece of evidence is that both my daughters and my ex-wife had a very high D-dimer test. My wife had a D-dimer level about 1.8, I think it was. In D-dimer testing, levels above 0.5 are of concern. So once you get above 1, they talk about hospitalisation. She was in a terrible way. She could hardly breathe. And so she started taking some of these combinations, and she's now got a D-dimer level of down to 0.25. So it's only anecdotal, but we're confident. My two daughters are now healthy and running their businesses and back to normal. They've just had recent blood tests. They're not showing any sign of some of the work you and I have done, Tom, with the blood separation. So I'm quietly confident that the combination they're taking has had benefits. But we can't talk about it because we're not medically qualified. We've got no science backing this at all, other than papers that do support us. And
well, it is a similar protocol to what Dr. McCullough's. He's doing nanokinase, bromelain, curcumin for his patients, and he said he'll be the first to tell you there's been no randomized control trials, but he says a lot of his patients are showing good progress with taking those drugs and those supplements in combination. And the beauty of that is you can order them, from Amazon. You can get them off the shelf. They're over-the-counter drugs.
Right. Okay, just two things before we finish. The blood test that you referred to, that you did, Greg, that is, if I understand it correctly, you have some blood drawn, like 30 milliliters or so, and put it into one of these little vials. and then bring it home, put it into the fridge for 24 hours, and then take it out and see whether the blood has separated into the plasma and the serum, correct? And there should be the red blood should be on the bottom and the serum should be on top. And there should not be some sort of white opaque clot swimming on top of the plasma, correct?
Correct.
Okay,
Yeah.
so in case anybody wants to do that
let's
test.
leave the conversation, if I may. I've actually got to go to another appointment. Tom, I'd like you to continue on with this, please, because we've done some good work in this, okay, and primarily at your instigation. So, sorry, Cornelia, could you excuse me, please?
Yes, of course. Thank you so much. You gave us a ton of time.
And thank you also for the opportunity. And I hope to maybe have another session with you a little later on when we've got some animal feeding results, because I think they'll be very interesting. But
Let's do that.
I'd definitely like you to continue on, please, with this simple blood test, because it's simple. It's simply a matter of having the blood taken home, put in the fridge overnight and observing. And we've got some very, again, anecdotal but interesting results. Okay, so I'll say abiento, everyone. And thank you very much.
Thank you so much. Thank you very much, Greg.
Thanks,
Bye-bye.
Thank you so much for your time.
Take care, Greg. Yeah.
All right.
And the next step that he's talking about is very important, because we want to see whether we can take some of the ground up sample of the white fibrous clot and then put that into solution, inject it into animals, test animals such as hamsters.
He told me about that.
rhesus
Yes,
monkeys,
he told
those
me
type
about
of things,
that.
yeah.
Right.
And see if there's this prion activity, right? If then that animal develops a white fibrous clot. In other words, is the white fibrous clot material contagious, infectious? Is it seeding? Does it then, implant itself in the vascular system and then grow and form a white fibrous clot to kill the animal? And we want to see if that happens, and if it does happen, how quickly does it happen? Because then that, if it does, that's, even more alarming, right? Because it proves the presence of prion material in these white fibrous clots.
Exactly. And he already said at the beginning of the interview that I think they're already doing these tests, right? But
correct. I've seen the results of one test, the very first test on a hamster, and it seems to confirm presence of prion material. I'll just say that.
Well, okay. So that's very, very interesting and important.
Yeah.
So yeah, let's take him up on his offer to do a second session.
I think so.
And,
yeah. In terms of the blood test, he's a few months back, myself, my assistant Laura Kasner, we both had our blood drawn. Laura was a little concerned because she's taken 2 Moderna shots. And then she stopped after that, but she took the first 2 Moderna shots. Now, to her credit, for the last 3 or 4 years, she's been on nattokinase, bromelain, curcumin, the regimen to, help break down clots or stop them from forming if if there's a chance. And I have never taken a COVID vaccine. So we both had our blood drawn, and the good news was our test tube showed basically the red blood cell layer at the bottom, dark red blood cell layer. Then there's a real thin buffy coat that sometimes you can or cannot see that contains your platelets and your white blood cells. It's almost invisible. And then on top of that is the plasma layer, which is kind of a yellowish colored liquid. And that is basically mostly water, but it also contains your fibrinogen, which we've been talking about. That's that protein that's in a soluble liquid form in your blood, in your plasma, that then converts into this white solid fibrin when the normal clotting process happens. But if abnormal clotting is happening due to the spike protein, phosphorylated spike protein, and the phospholipid nanoparticles combining with your fibrinogen to form these bizarre white fibrous clots, that can precipitate out in people that are having problems in their vascular system in their blood sample. And you'll see sitting at the bottom of that clear yellowish layer of plasma, you'll see a white clot in some people. And
I see. Yes, I've seen that. I've seen the picture in one of the papers from Dr.
Burkhart. Yes.
Yes, exactly.
Yeah.
It's a great picture. I'm going to put it into the interview as an overlay because you can very clearly see this.
That's
this
where I
white clump.
first noticed the white clump too. Yeah.
Yes. Tom, how did you get someone to draw your blood and then take it home?
I prefer not to say. I don't want to get the doctor in trouble that did that.
Oh, okay. Because I think that's a problem.
Because,
you cannot. I mean,
yeah,
I don't think I can. Yeah.
I tried, by the way. I first here I live in Dayton. I'm a retired U.S. Air Force major. So I went to my base hospital. I asked him, hey, I went in just the blood clinic, and there was about 3 or 4 ladies in the room where you sit down in the chair and you offer up your arm. And I said, I'd like to have my blood drawn and actually be able to take home my tube. And she said, well, I don't know if we can do that because usually they take the sample, then we send it off to lab for analysis, right? She said, let me go check with my boss. Well, the boss came in, and the boss escorted me across the hall into her office and asked me why I wanted this blood sample for myself. And I explained, I was truthful to her. I said, hey, we're investigating the potential of these white fibrous clots that are forming inside of people. We believe that there's a simple blood test that can be done to determine whether or not you may or may not have this material in you. Just a simple blood separation test where you let the blood stand in a vertical tube for 4 hours overnight, whatever, until that blood separates out. And then you just visually look and see whether there's white clot material in the bottom of the plasma layer. And she said, I'm sorry, sir, but we can't let you do this. I said, well, why not? It's my own blood. And she said, it's a biohazard to let you walk out of the hospital with your own blood. And I'm thinking, what are you talking about? People skin their knees all the time, right? And bleed and stuff. And you
Yes.
clean it off, and you throw your paper towel away that you bought your blood with. Is that a biohazard too? What's going in the way?
Well, Tom, Tom, they should not have let you into that clinic because you are a walking biohazard. You can start bleeding at any time. But that is a real problem. Did she ask you whether you are a vampire?
No, but I can see her eyes got a little more saucery when I explained what we were doing, that we were, because I've done these surveys with finding a large percentage of corpses of white fibrous clots, and we believe there's a problem in the blood. So I kind of got the impression that the people in that office kind of knew that the blood has changed over the last few years. They never said that, but I kind of got the impression that when I mentioned this to her, she wasn't totally surprised by what I was saying, as if, they've been checking a lot of blood samples in the Air Force Base Hospital over the last 4 years. And I don't know what they're doing, or whether they're just leaving the problem alone, or whether they're secretly doing testing with it or not. I have no idea, but I'll just tell you, I walked out of the hospital that day with no sample.
Right. Yeah, okay, but which is a real problem, because I guarantee you that this will happen in 99% of cases in all likelihood.
If people will try to go there and get their blood drawn and want to be,
they will not be willing to give you your blood. So it's a simple test, and you cannot do it. I mean, unless maybe you
have a friend that knows how to draw blood, you happen to just know a nurse or somebody who works in a lab like that that takes lab specimens who could do it for you.
Yeah. Well, it's pretty atrocious that
it really is.
You're not allowed,
because that's one of the things when I first discovered this. And another thing I did, by the way, is I called my assistant Laura Kasner, got a hold of I think it was a post on one of Steve Kirsch's Substacks from a woman that said she went on a bus called the Big Red Bus, and it's run by a company called One Blood. And One Blood, it's similar to the American Red Cross. They go around on their big red bus, and you can go on board and donate blood right there on board. Well, this woman had put in a Steve Kirsch Substack post that she had been asked a question before she gave blood. One Blood wanted to know, had she been COVID vaccinated recently? And she said, oh, that's an interesting question, that they asked her pre-donation. So I went ahead and I contacted their headquarters of One Blood. I talked to a nice Jamaican lady there named Rose, and I asked her, why does One Blood ask this pre-donation question? And she said to me, One Blood only accepts the donated plasma from unvaccinated donors. If you've been COVID vaccinated, they will not accept your plasma. And so my next question, well, why is that? I said, and then she went kind of silent on me. I said, well, is it because possibly the mRNA from the vaccine is in the plasma? And then she said something to the effect, you're a pretty smart guy, aren't you? So that was so that was disturbing. And what was interesting about that is Clayton Morris from Redacted News, who I was in touch with, and we did a interview about this. Clayton himself contacted or looked at the website of the American Red Cross and saw that their policy was they will accept both the donated whole blood and the donated plasma from COVID vaccinated donors. However, they want you to wait at least 2 weeks before giving blood after your last COVID vaccination. They don't want you to get a COVID vaccination and then give blood right away. And remember, one of the things that they told you at the very beginning of the pandemic was when you got the COVID shot, that mRNA is supposed to dissipate within 2 days to a week, right? So it's supposed to leave your body after a week. And you're not supposed to
They still say that. I checked some websites yesterday to see what people say, or what big medical companies, etc.
Well, we know from scientific papers that's not true.
They still say, well, of course we do, but they still say that. They still have that old information that is completely outdated and completely misleading. But this is one really important topic that we didn't even talk about with Greg. What impact does that have on the blood supply, on the blood banks? How can contaminate? I mean, blood that it contains these amyloids or these clots, and the possibly prion-seeding activities, amyloids with prion-seeding activities. What if you get that in a transfusion? I mean,
Right.
Also, I would have liked to go further with him into the whole shedding question, because he mentioned the exchange of fluids, but there's also some evidence. I think Dr. Corey did a little bit of research on that, on shedding, that doesn't involve the exchange of bodily fluids. So that would have been, it's another interesting.
Exosomes, basically, that you can breathe out. You can sweat out the mRNA or the spike protein, or the phospholipid nanoparticles can come out of your body in different ways, including your breath. So it's definitely an area of concern. Now, the good news is if a person's not taking the vaccine and they have been shed on, it's going to be more difficult for that spike protein to get inside your vascular system and do damage. Because first of all, it has to fight through your mucosal membrane. It has to get, from the outside of you, inside of you. It has to go through your nose, your lungs, and your protective mucosal membrane, whereas the vaccine gets directly injected inside you. So it bypasses the mucosal membrane and can get directly into the bloodstream.
Although, they are now developing mRNA COVID shots that are administered as a nasal spray.
Yeah, and I'm not sure how effective they're going to be, because like I said, those will have to go through your mucosal membrane as well. So I don't know how they're going to be... how they're going to go in terms of damage compared to the intravenous vaccines that are used with injections. Another thing that was always talked about was the aspiration of the vaccine as well, right? They decided not to aspirate, during administration.
Why was that? Why did they decide not to aspirate?
I think they're people might have to get stuck twice, maybe, and they didn't want people to have to get stuck twice. If you stuck the needle in
that doesn't make any sense.
I know it doesn't, because
that was never a reason against it. And of course, you don't want to shoot it right into the
vein or artery,
system, right?
Yeah.
You want to put it into the muscle.
Yeah, but even when it does go into the muscle, I think there's been studies that show about 70 to 75% of the mRNA gets picked up by the bloodstream anyway. It leaches into the bloodstream, into the lymph nodes. So
it does. Yeah, it does.
So, but I do know that there are a lot of people that they said that when they got injected accidentally into a vein or artery, like they very quickly knew what was going on, because they had like a metallic taste in their mouth, and then started suffering very quickly some of the adverse effects from the vaccine. So,
well, I'm looking forward to the next interview with Greg. To hear more about the experiments on animals, what happens with them, what happens when they are injected with these prions. So a lot more things to find out. Okay, thank you so much.
Thanks for having us, Cornelia, and we will get back to you when we've got new and fresh information. This is a very serious issue, and like one last plea: universities, FDA, CDC. Get on the ball and start investigating this, because this problem is not going away.